In this video, we'll discuss the role of phylogenetic trees and their use in examining evolutionary relationships between organisms. The use of phylogeny in examination of the fossil record provides independent and corroborating evidence of evolution, which we will examine by looking at the phylogeny of human evolution. Remember from the last chapter that distinct populations will separate and diverge again and again throughout time, giving rise to multiple descendant species. This history of descent with branching is called phylogeny and can be diagrammed using a phylogenetic tree. As species proliferate, their evolutionary relationships to one another unfold in a tree-like pattern, with present-day species as the tips of the branches here, and their last common ancestors at nodes from which they branched off. This means that node 1 would represent the original population, while node 2 would represent population B, which had branched off into B and C. The branching order found in a phylogenetic tree hypothesizes the evolutionary relationship within a group. When examining the tree, two groups are considered to be each other's closest living relative if they share a common ancestor that is not shared by any other group. These are called sister groups. For example, frogs and salamanders would be considered each other's closest living relative and would make up a sister group, because they share a common ancestor here that is not shared by anyone else. You do not need to look only at single branch tips when using phylogenetic trees. For example, the closest living relatives of the entire tetrapod group would be the lungfish, because the group of tetrapods share a common ancestor with the lungfish here. The closeness of the relationship between groups is determined by looking to see how recently the two groups share a common ancestor, based on the length of the branch lines, which represent evolutionary time. So frogs and salamanders are much more closely related than lungfish and tetrapods. Examining phylogenetic trees lets you observe the evolutionary relationships of organisms. In order to do this, you must examine monophyletic groups, such as the amphibians shown in blue. Amphibians here are a monophyletic group because the group includes a common ancestor here and all of its descendants. Evolutionary relationships are more challenging if you are studying a paraphyletic group or a polyphyletic group. A paraphyletic group, such as the reptiles shown in green, is a group that includes the common ancestor but only some of its descendants. For example, birds are not part of the evolutionary group, but they are not reptiles. Polyphyletic groups, such as bats and birds, do not share a common ancestor. We construct phylogenetic trees based on a comparison of the anatomical, physiological, or molecular features that make up an organism. Common features, called characters, can arise in organisms for two reasons, either through common descent from a shared ancestor, called homologous traits, or through convergent evolution, in which the trait evolved independently in two separate groups, called analogous traits. In this phylogenetic tree, mammals and birds both produce amniotic eggs, and both diverge from a common ancestor. Thus, we hypothesize that this is a homologous trait because birds and mammals each inherited this character from a common ancestor in which the trait first evolved. This is different than wings, which be an analogous trait, because the evidence for the tree supports the view that wings in both birds and bats do not reflect descent from a common winged ancestor, but rather evolved independently in two separate groups. Fossils are the remains of once living organisms, preserved through time in sedimentary rocks. They allow us to determine the timeline of phylogenies, provide a record of extinct species, and place evolutionary events in context within the Earth's history. In order for an organism to be fossilized, it must first be buried, after which sediments accumulate over time and harden, as can be seen in the layers of the Grand Canyon. Most fossils preserve the hard parts of an organism, those features that resist decay after death. Organisms that lack hard parts can leave a fossil record in two other ways, though, through trace fossils, such as tracks and trails, and molecular fossils, such as DNA and proteins. Because of the lack of soft tissue in the fossil record, some aspects of biological history are preserved poorly, while others, such as the forms, functions, and diversity of skeletonized animals, are preserved well. Geologists recognize that groups of fossils changed systematically from the bottom of a sedimentary rock formation to its top, and that fossils that occurred in the layers that lie on the bottom are older than layers at the top. This pattern of bottom to top allows geologists to map out the series of time divisions that marked Earth's long history in the geological timescale and record the evolution of life on Earth. While the layers of fossils in sedimentary rocks can tell us that some fossils are older than others, they themselves cannot provide an absolute age. To do that, we use a process called radiometric dating. Radiometric dating uses the radioactive decay of an isotope to map the time scale of a substance. The most commonly known radiometric dating is carbon dating, which uses carbon-14 isotopes generated from cosmic radiation in the atmosphere. C14 is incorporated into carbon dioxide, which is then incorporated into plant material through the Calvin cycle of photosynthesis, and then animals when they eat the plant material. After death, the unstable carbon-14 begins to break down, losing an electron to form nitrogen-14, which is a stable isotope of nitrogen. C14 breaks down at a constant rate called the half-life, where half the isotope is converted in 5,730 years. So by measuring the amount of C14 in decaying material, you can determine how many years ago that organism died. 5,730 years may sound like a long time, but is actually a relatively short half-life, which makes C14 dating only useful if dating material is younger than 50,000 to 60,000 years old. Archaeologists date older samples by looking at the radioactive decay of uranium or lead, which have much longer half-lives. Phylogenies hypothesize impressive morphological and physiological shifts through time, amphibians from fish or birds from dinosaurs. In fact, phylogenies based on living organisms generally place birds as the sister group to crocodiles and alligators, but birds and crocodiles are obviously very different from each other in structure. So does this phylogeny have supportive evidence in the fossil record? The answer is yes. In 1861, a fossil was discovered of an organism that lived 150 million years ago, whose skeleton shares many characteristics with small, agile dinosaurs, but several of its features, including wing-like forearms, are bird-like. The fossil shown preserved evidence of feathers. These types of fossils that show transitional ancestors between modern groups are called transitional fossils. Data from phylogeny and fossils are often used together to show strong evidence for evolution, because the phylogeny makes use of living organisms, while the fossil record supplies information about dates, environmental context, and transitional common ancestors. We have used phylogeny, looking at comparative anatomy and molecular analysis, and the fossil records to construct human evolution. Comparisons of DNA sequences suggest that chimpanzees are our closest living relative, and structural observations show that chimpanzees and humans share many features, but also have key differences. There are about 400 species of primates, all which share certain features that distinguish them from other mammals, such as opposable thumbs and nails instead of claws. Primates are further subdivided, and one line of Old World monkeys gave rise to apes, which lack a tail and show more sophisticated behaviors than other monkeys. Apes are split into the lesser apes and the great apes, which have more robust canine teeth, including the orangutan, gorilla, chimpanzee, bonobo, and humans. The common ancestor between chimps and humans, here, would have been found on the planet approximately 7 million years ago. During that 7 million years, the fossil record shows some of the changes that occurred leading to modern-day humans, such as the ability to walk upright, which evolved before the increase in brain size seen in humans. It is important to recognize that humans did not evolve from chimps, as is often stated, but that humans and chimps both evolved from a common ancestor that was shared 7 million years ago, so we are very, very distant cousins. The evolution of modern humans, called the hominin lineage, produced many different species in Africa, and in fact, were often several different species living at the same time, as indicated by the overlap in red lines seen here in the graph. All hominins have a common ancestor, but not all of them led to modern humans, called Homo sapiens. Instead, many of these branches ultimately went extinct. The fossil record indicates that the first hominin lineage to venture out of Africa was Homo ergaster, here, approximately 2 million years ago. Ancestors of Homo ergaster in Europe are associated with Homo erectus, and paleontologists debate if they are the same species. While those of the Homo ergaster lineage that remained in Africa became the descendants of Homo heidelbergs, here. These are the likely most common ancestor of Homo neanderthals and Homo sapiens. Homo heidelbergs migrated out of Africa approximately 600,000 years ago and eventually evolved into Homo neanderthalis, which first appeared in the fossil record in Europe approximately 400,000 years ago. Homo heidelbergens that remained in Africa became the descendants of anatomically modern Homo sapiens, which first appeared in the fossil record about 300,000 years ago. And they themselves migrated out of Africa approximately 100,000 years ago. While Neanderthals were thicker boned than us and had flatter heads, their brains were about the same size as ours, and they coexisted with Homo sapiens for approximately 5,000 years. They disappeared from the fossil record approximately 30,000 years ago, although modern genetic evidence indicates that there was interbreeding between Homo sapiens and Homo neanderthals during the time they coexisted in Europe. Many modern-day humans of European heritage contained 1 to 2% Neanderthal DNA.